So thank you very much for attending. My name is Ann Nielsen, and Rutgers University is pretty much as far away from Hawaii as possible, but we actually have a research project that we're going to be starting in Hawaii on coconut rhinoceros beetle soon. So I'm excited to come and visit you guys. As Rashawn said, I've been studying brown marmorated stink bug for quite some time. I think I started around 2003. I did my PhD on brown marmorated stink bug. It's even hard for me to say. And then I did a couple postdocs and then came back to help tackle some of the problems. And we've done everything from basic biology, organic pest management, to management and specialty crops, to genetics work. So what I'm going to give you guys is an overview of brown marmorated stink bug biology and what I think are the keys to understanding its biology and how to implement management, or at least what's worked for us on the mainland here for management if it becomes a problem in Hawaii. And all right. So brown marmorated stink bug, it is an invasive species from China. It started off in the eastern Pennsylvania before spreading to New Jersey in a few years after that. And we believe it came over in shipments of packing material in eastern Pennsylvania. It overwinters as an adult stage, which is pretty common for stink bug species, but this particular species has a tight association with human-made structures. So it also serves as a nuisance pest in many areas because it likes to overwinter inside human-made structures like homes, as well as barn buildings and natural areas such as dead upright trees or cliff outcroppings. Now that's in areas where it overwinters, and I don't know, we don't have a lot of data yet on how the biology of brown marmorated stink bug in tropical areas. But for us, it does have a facultative diapause stage, and we have 2 generations per year. So the adults come out of overwintering as indicated here. They lay eggs. There's a slight gap between the time the females emerge from overwintering and the time that they become reproductively mature. They lay those eggs starting around mid-May in this area, and then they go through the 5 nymphal instars before becoming adults, and they can continue this life cycle again when they have 2 generations per year. Then they will again seek out an overwintering site and spend the winter there as well. So the second instars through the adult stages, which is the bulk of their life stage, can cause feeding injury on multiple specialty crops as well as field crops and nursery crops. This is a highly mobile pest. The nymphs themselves can move pretty rapidly to different host plants, but the adults can move as many as an average of 5 kilometers in a single day, with some super flyers traveling 50 kilometers in a single day. So these factors, combined with the over 170 different host plants this bug has, make this a landscape level pest. It has a global distribution. Now, most of this is invasive. The black area, as indicated on this global map, is its native range. And then this is just kind of a series of timelines of when it became detected in different areas of the world. You can see that it was first detected in the United States, and then there was a big gap between 1996 and 2007 when it was first detected in Switzerland, and then it was found in Canada, and then multiple locations in Europe. And most recently, it was found in Santiago, Chile. But as of that, we have had detections in New Zealand, as well as Australia, but we don't consider detections on this map. We're only considering established breeding populations. And there's differences between the pest status in different parts of the world as well. However, because of this overwintering habitat that creates a nuisance factor for many people, I particularly don't mind if there are stink bugs in my house, but the general populace doesn't particularly like having stink bugs flying around their homes, and they do fall into food and are a nuisance, and they buzz around your TV and such. As you can see in this paper, this article published on the front page of the New York Times, these homeowners are literally sweeping brown marmorated stink bugs off of their porch. And so you can have thousands and thousands in your property. The highest recorded by a scientist was over 52,000 brown marmorated stink bugs overwintering in his house. We see a similar situation in Italy. All these black spots are brown marmorated stink bugs seeking overwintering sites. Unfortunately, this happens to be at a bed and breakfast in Italy. So it's not a very good thing for their tourism industry as well. I've done a lot of work with biosecurity in New Zealand, and they are very concerned about brown marmorated stink bug coming in and impacting their horticultural industry, specifically the apple industry. Now, there's been a lot of different population modelings, both Climax and niche modeling, to try and figure out the potential distribution of brown marmorated stink bug based off of where it has been found in both its native range in Asia as well as the United States. However, all of the modeling that's been done, there's a lot of difference in the models. For instance, some models say that New Zealand is a really good source for them, and other models will say that only the North Island of New Zealand are good sources for them. However, none of these models have really included Hawaii. So I wanted to make sure to include some of the other Pacific Islands in here to perhaps give you an idea of what might happen, but there is not a lot of information on the potential distribution for Hawaii. I will say, however, that these potential distributions that we are seeing in Mexico and Central America and Cuba and Haiti have yet to occur. We are not seeing populations of brown marmorated stink bug in high numbers in any kind of more tropical or subtropical regions. And that also extends to the state of Florida and even Southern California. We have populations in Florida, but they're not really large populations and they're not causing significant damage. We are seeing populations in Central Valley, California, which I'm sure Surendra will talk about or might talk about a little bit, that are causing some problems, especially in almond crops and peaches. So the current distribution of brown marmorated stink bug right now, we update this map once a year, it's been found in almost every state. The states in green, including Hawaii, are where we've detected BMSB or intercepted it. That does not mean that it is a breeding active population. Those we consider a nuisance problem only. So you can see
Joe.
Join the meeting.
Of the United States is mostly green, where we're not really seeing problems with that. Most of the issues that we're having in terms of our agricultural and nuisance problems in the United States are still centered in the mid-Atlantic states over here where I'm located, as well as in the Pacific Northwest, specifically in Oregon. However, we did have outbreak populations of brown marmorated stink bug starting in late 2009, but primarily in 2010. This was devastating for our tree fruit growers specifically. We had about this peach grower, for instance, abandoned his entire crop this year. If you look closely at these pictures, you can see dimpling. I don't know if you can see my cursor or not, but you can see dimpling on these peaches and depressions and rot. Those are all causes of brown marmorated stink bug feeding. This is a piercing sucking insect. So as they're feeding, it's creating corked tissue that becomes deformed as the fruit develops. Brown marmorated stink bug in peaches is especially bad because they feed on this crop throughout the entire growing season of the peach. These peaches here are well before pit hardening, so they're still, yay big. These brown spots and even this green spot are the corking that we see from brown marmorated stink bug feeding very early on in the season. So this is in the first couple, late May, early June, that we see this type of injury. And the corking occurs because as the stink bug is feeding, it's releasing digestive enzymes, and that kills the surrounding cells of the fruit. And during this stage specifically, they're very mitotically active. And so that fruit then kind of grows around that wound spot that's now dead. And then you can get this type of defaced or cat-faced injury that you can see. This also occurs later on in the season, and even sometimes this can kind of heal over and we get almost hard calcifications inside the fruit that are kind of very gritty. So on average, we had about 60 to 90% of losses in the peach industry in the mid-Atlantic states in 2010, despite intensive insecticide applications. We had a similar story in apples. In apples, it's primarily a late season problem, and you can get these dark and sunken areas on the fruit when you peel that fruit, you can see a lot of corking injury on the inside, to the point that we had fruit that was rejected from processing because they couldn't peel the fruit. So this resulted in $37 million in losses to our apple industry in the Mid-Atlantic state alone. Even in 2013, after that peak population injury, we are still seeing between 4 and 44% of injury in New Jersey peaches as of 2013. I think we're down closer to around 4 to 10% injury now. But what's interesting is that although BMSP populations on a kind of national scale started declining after 2011, 2012, we're starting to see an uptick again in our populations in the Mid-Atlantic state with associated increases in injury as well. So why is BMSB such a bad pest? It's an aggregated feeder. They do have an aggregation pheromone, and they have an aggregated distribution in the landscape. So you may have a problem on one part of your farm and not a problem on the other part of your farm, or your neighbor may have a problem, but you don't. So the populations are very patchy within the landscape. They do kind of feed in aggregation, especially under high densities, and they have a longer proboscis or stylet than a lot of native stink bug species that we have. So they can kind of get deeper into that fruit tissue. They're pretty reproductively fit. We have females that have about an average of 200 eggs per female, with some females laying as many as 300 to 400 eggs. It takes them about 538 degree days base 14 for development, or 1,000 degree days base 57 Fahrenheit for development, which allows them to have 2 generations per year, but those generations overlap, and I'll get to that in a second. So as many invasive species, as you are all familiar with, we have an escape from natural enemies. We only have about 1 to 2% of our eggs that are parasitized by egg parasitoid species, which are predominantly in the either Anastatis genus or the Celeonid family. We have about 10 to 15% predation by generalist species, which is pretty good considering this is an invasive species, but it's not enough to kind of tap down to the level of economic control that we need. In many ecosystems, this becomes the dominant stink bug species, and not because it's out competing, just because it's so abundant. It is a continuous pest in fruit production, and as I mentioned, because of this multiple utilization of host plants throughout the growing season, they will follow host phenology and host development, and so they kind of hop from the forest to the peaches, to the wheat, to the apples, to the corn, to the soybean, to the vegetables, to the wine grapes, and then back to the forest systems again. So they kind of create this kind of hop, skip, and a jump throughout the landscape, at least in the Mid-Atlantic states. We have created a kind of high, medium, low, red, yellow, green stoplight kind of approach for specialty crops at risk to brown marmorated stink bug injury. The key things on here are apples, Asian pears, beans, edamame, eggplant, pear, hazelnut, peach, and corn in the high risk category. The low risk category, a lot of these we just don't know, or they're landscape ornamental plants. I would say though, we are seeing populations really high levels in almonds and also in kiwi. So I would move those up to the higher risk plant. When we first made this in 2015, we didn't have data on some of those crops. So in terms of Hawaiian agriculture, I think the 2 crops that are at highest risk are macadamia nut, just because of its association with both hazelnuts and almonds, and possibly coffee. There are other stink bug species that are pest in coffee. They feed on the berries early on, either transmitting fungi, or they cause berry abortion or give it off taste. So I don't know if brown marmorated stink bug would do the same thing, however, but that is a crop I would survey for. However, papaya and pineapple I don't believe would be key hosts, because they're not in any host plant families that brown marmorated stink bug currently utilizes right now. So the next things in terms of the critical things to know are about the population dynamics. So I've mentioned that we have 2 generations per year. We've done a lot of phenology modeling and developmental models for this pest. Early season populations are really low. And in some locations, people think that they don't have 2 generations per year because they're not catching nymphs, or they're getting very few adults. And so they think the pheromone doesn't work. That's not the case. What we have is overlapping populations for all of our generations. And if you look at our population model, that becomes a little more clear. You have our black is our overwintering adults, and red is the F1s, and 2 is the F2 adults. And so you can see, especially in the summertime, we have very strong overlap between F1 and F2, or first and second generation adults. And we can even have some of those overwintering adults survive through August. So this is a really long-lived species, and they will reproduce throughout their entire life cycle, which creates this overlapping generations. The populations do peak in mid-July, mid to end of July around here. And people often thought that was the F1 generation, and it turns out through our modeling that that's actually the F2 generation. So it's kind of fun to keep learning as we go along. Here's just kind of a closer up picture of what those overlap populations look like in my geographic location, where we have our overwintering populations, our F1 adults, and then our F2s. And so this big peak occurs at the end of July here. We do see dispersal into fruit crops, specifically in the peaches, around 170 degree days, base 57 Fahrenheit. So it's and as they're moving into peaches from the woods or from their overwintering sites, they are mating almost immediately. And will lay eggs, and those nymphs can feed on peaches as they develop. So the orchard crops that are at highest risk are peaches and apples and any tree fruit that borders woods. And that's the way our landscape is made up. We have a lot of wooded habitat and then the farmland surrounding that. So it's a very patchy kind of network of landscape here. But this movement throughout the landscape requires intense repeated insecticide applications. As of right now, insecticides remain our primary tool for management. And unfortunately, when BMSB first came out, this caused basically a 4-fold increase in insecticide use. And this disrupted over 40 years of IPM programming that we had. It caused growers to abandon a lot of our more softer behaviorally based management tactics. And because the insecticides are primarily pyrethroids and neonicotinoids, it caused some major problems with secondary pests such as mites and mealybugs and aphids. So we needed to figure out ways to combat that. So we do have an aggregation pheromone trap, which I'll get to. We have a phenology model to predict life stage occurrences. We have a lot of different management tools that we've put together over the years. I'm going to talk about these ones here in bold, specifically border sprays, trap-based thresholds, and biological control. Our aggregation pheromone is commercially available. It's produced by at least 2 companies, Trace and AgBio. It's a 2-component aggregation pheromone plus a synergist. So we have two different aggregation pheromones, a major and a minor component, and then we use a chiromone of a native Plaudeus stali, which is a stink bug found in Asia. And that actually synergizes the BMSB pheromone. And this MDT is what we used during our early studies. And we would only get late season capture. So these epoxy bisboline components allow us to get early season capture as well. But this synergizes it together. So you need both components for monitoring. We've developed 2 different types of traps. We started off with this black pyramid trap. It's very, very effective. It's a trunk mimic. The insects fly into it, they crawl up, they get captured into the top. However, it's about $30 a trap. They get hit by mowers and other tractor equipment pretty frequently. And so we've kind of switched to this clear double-sided sticky panel trap, which is still baited with the pheromone lure here, as shown as this black strip. And we just put them on a 5-foot tomato stake. So they're about 4 feet in the ground. And we change the clear sticky traps as needed. And there's a really strong correlation with these 2 traps. We get more with the pyramid trap, but we think that the clear sticky trap is good enough for management decisions. And the traps need to be placed at the crop edge or neighboring wood edges. There's some concern about putting the traps in the middle because it's an aggregation pheromone trap that it will lure the bugs into the middle of the crop. However, aggregation pheromones work really differently than what we're kind of more used to with our sex pheromone traps. So we have our aggregation pheromone trap here on the left and a sex pheromone trap that we would use for like a moth pest on the right. And the area of attraction or the area of response is really different between the two. So with a sex pheromone trap, we're only attracting the males and it's a point source. So we're attracting them to a very specific location and we don't get any increased injury or anything like that in our crop. However, with an aggregation pheromone, it's a very large area of response and it's attractive to all life stages. In the case of brown marmorated stink bug, it's attractive to males, females, and nymphs. So it's a really good tool for that. However, we do get spillover effects of feeding injury because they don't all go into the trap. So we know that we do get reliable adult and nymphal captures with these trace lures on the sticky traps across different densities of BMSB. We've tested this on a national scale. And so at high, low, and medium densities, we get very consistent trapping. You don't want to hang them in the canopy of the tree though. They get distracted too easily. And we can get about 4% of the population in about 4.8 hectares in 12 hours. So we do get about a 4-meter spillover effect in peaches. So you can get injury on neighboring trees in a peach crop. And it's a little less of that in apples. So we've developed a lot of perimeter-based strategies, some with the aggregation pheromone and some without. So we know from other research that I've mentioned earlier that BMSB is a landscape pest and it's often moving from one crop to the next or from the wood edge to the crop. And as it does this, we've been able to identify through multiple methods that they actually arrest their movement on the perimeters of the crop. And this creates a very strong perimeter-driven or border-driven injury and populations. So we've developed a couple different tactics to manage this. From my lab, we've developed border sprays, which is exactly what it sounds like. We're exploiting the perimeter-driven dispersal behavior. We typically do these every week, which is a lot, I know, but we're spraying the perimeter of the orchard, not the trees surrounding it, the actual orchard itself, and the first full row. When we do this, we can apply about 25% of the insecticides that are applied with grower standard management programs and get the same level of injury at harvest. So we're saving the growers money and we're saving insecticide application. And we've some evidence that this also conserves natural enemies. Work led by my colleague Tracy Lesky at USDA uses an attract and kill approach where they put high doses of aggregation pheromone on the orchard borders at select locations, and then you treat only those select trees. So you have a spatially refined insecticide application. And again, we get the same reduced, same or equal injury at harvest as the full block sprays. But you do get a lot of injury on those baited trees as well. So for the border sprays, we've actually done this up to 20 acres. We've done this in multiple different habitats. And this was grower-driven data. So there's a lot of variation here, but essentially we've been doing this since 2012. But when we go up to 20, we can go up to about 10 or 20 acres in size before we see that border effect starting to fall apart a little bit. So that's an additional cost savings to the grower. Tracy Lesky's lab wanted to compare both attract and kill, perimeter-based or border sprays, and then a threshold that they've developed using 10 bugs per trap, a cumulative number of 10 bugs per trap. And so what they did is that they tested different spray intervals. I mentioned that our border spray approach treats every week. So they wanted to try and extend that to 7 days and 14 days. And then they did the attract and kill at 7 days and 14 days. And then they compared this with their treatment threshold of 10 bugs per trap. And they did this on commercial farms, and they were able to compare this with a no insecticide control. So grabbing a table of the results from her study, what you can see is that you do get significantly lower injury in the fruit, both on the perimeter and the exterior. the perimeter and the interior, sorry, in the perimeter-based control methods and the threshold treatments. However, in the perimeter control, you do have a increase in the number of insecticide applications, full block insecticide applications. So there's some trade-offs there. But this is really encouraging and can lead to some additional questions on maybe refinements of the threshold or incorporating the threshold and the perimeter-based management together into a system so that we can further reduce our insecticide applications while kind of creating these more eco-friendly orchard systems. And I know I'm talking about insecticides only, but that has been our growers' primary defense against brown marmorated stink bug at the commercial level. However, we do have some natural enemies. The enemy release hypothesis suggests that is a strong reason for success of invasive species because they arrive here without their native predators to keep their populations in check. We know that natural enemies are important regulators of populations, both economically and numerically. And we have the assumption that a lot of our endemic or native species will not accept the invasive species. So there's some, Amanda Koppel did work on native stink bugs and found that they had 67% parasitism in field crops and less than 1% of predation in native stink bug species. When we've done big surveys on brown marmorated stink bug in organic farms using sentinel egg masses, as shown in this picture here, we found less than 1% parasitism, which was really discouraging, and between 3 and 20% predation. And that varies a lot depending on how you classify predation. But so we're kind of seeing a switch. It's a flip-flop of what we would expect from our native species. And we did this by both monitoring with sentinel egg masses, and then we've also switched to monitoring with yellow sticky cards for other natural enemies. We did do some laboratory microcosm works with our native predator species to determine which life stages of BMSB different predators would attack. And so we have our damsel bugs, our Nabidae here, we have assassin bugs or regiviids, pterygonoids or katydids here, and of course our coccinellids. And what we found was really interesting. Our nabids prefer our second instar nymphs. The assassin bugs prefer our first instar nymphs, as you can kind of see this one munching away on one right now. Our katydids are really good egg predators. They will consume the entire egg mass if given enough time, to the point that it looks like it's missing. And so a lot of our missing sentinel egg mass data we account towards katydid predation. Coccinellids were not great predators of BMSB, but when they did feed, they did seem to prefer the second instar nymphs. So we do have a little bit of variation there with the different coccinellid species as well. So I mentioned these different management programs, both our border-based sprays and our attract and kill and threshold treatments. In all of these studies, we've also looked at the impact of natural enemies under these different IPM tactics, because we want to make sure that we don't have outbreaks of our secondary pests, and we're not using chemicals that are harmful to our natural enemies. Well, we're limiting the impact on the effect of our natural enemies. So we do a lot of this monitoring with both sentinel egg masses and then again with our yellow sticky cards. When we look at our border spray approach, we see that we have a significantly higher number of parasitoids on our yellow sticky cards in orchards that use border sprays than the orchards that are using our standard insecticide sprays. And we actually have recovered Trisulcus japonicus in many of these orchards that are using the border spray approach. And Trisulcus japonicus is also known as the samurai wasp. It is the co-evolved natural enemy or egg parasitoid of brown marmorated stink bug in Asia. And we have adventive populations of that here in the US. And it seems to have arrived multiple times because there's a lot of genetic differentiation between the populations throughout the US. When we've done mass rearing experiments and release of Trisulcus japonicus in orchards, we do see that they will disperse to peaches. This has predominantly been thought of as like kind of a wood associated or arboreal linked species, but we can get them to disperse into peaches as far as 35 millimeters. However, like many parasitoids, they're very sensitive to insecticides, especially ones like Entrust or Delegate. And so we have to kind of figure out how we can incorporate our natural enemies and our IPM tactics. So Tracy Lesky's lab led some work where they took these different border spray approaches and then alternate row middles, which is the way we tend to manage our crops, which is alternate. Every other row is sprayed every week. And they got very strong parasitoid emergence of Trisulcus and in both of these treatments. And it was similar to untreated egg masses that they put in the woods. And we can also create refugia with the use of insectary plants as well. However, Trisulcus is not cleared for release by the USDA, so you can only do studies on it with populations that you find, detect, and rear it within your individual state. So to summarize, BMSB has a really large host range, over 170 different host plants and landscape level impacts. It's hard to predict if it's going to be a problem in Hawaii, but I would guess based off of its host associations that macadamia nuts and coffee are probably the highest risk. If it does become established, it would be at least bivoltine and have a similar phenology as observed in the US. However, our populations will go into diapause based off a photoperiod, and we're not seeing that in some of the more tropical areas with warmer winters. So you may have year-round problems with it. However, the perimeter-driven behavior of BMSB can be exploited to improve agro-ecosystem health. And we know that TJ is here and is giving us a little ray of hope. So, you know, after 20 years of studying BMSB, we finally have some answers. So with that, I need to just thank a lot of other people, and I think my time is up.
Thank you, Anna. So we will have a question and answer session after Christina Mogren's talk. Thank you.
Okay, thank you.
Yeah.
I'm going to stop sharing.
So our next talk is from Christina Mogren. Christina, are you here?
I'm here.
Okay. So Christina Mogren is assistant researcher at Department of Pests. And I think she's research is focused on pollinator nutrition and the influence of landscape on pollinator health. And also she likes to talk about integrated pest and pollinator management, right? IPM. And so she did her PhD from University of California, Riverside. And I think she went on to do postdoc in University of, I forgot.
First postdoc was with the USDA in South Dakota. Second one at LSU's Ag Center in Baton Rouge.
Okay, then I think she joined the Department of Pests around 2017. Is that right? Okay, so today she will be talking about old and emerging threats to honeybee in Hawaii. So Tracy, I will make you co-host.
Okay, can you guys see? You can see my share screen though, right? All right.
Yes. Okay.
Perfect.
Yes, please.
Okay, great. Thank you. Thank you everyone for coming today, and thank you Roshan too for giving me an invite. I honestly, so I am an assistant specialist of pollinator ecology here at UH Manoa, and the longer I'm here, the more it becomes so clear that beekeepers and those dependent upon bee services are a really underserved community here in Hawaii. So I'm excited that I have the opportunity to kind of speak about some of the greatest threats facing this, relatively important commodity, being honeybees here in the state. So I've titled my talk Old and Emerging Threats. So I realize this is an invasive species conference, but interestingly enough, the greatest threats to honeybees in Hawaii and then also worldwide have already been introduced into Hawaii, despite very stringent measures that the state has taken to limit introductions of pests specifically for the beekeeping industry. So I'm going to talk a little bit about both of these today, and then also kind of my impressions on ways that the state and our current paradigm about how we treat pollinators in the state kind of needs to shift in order to make sure that these threats do not become more widespread, and then also do not spread to some of the outer islands where they're currently lacking. Oh, hang on. There we go. Okay, so honeybees have actually been facing unexplained declines for the past 150 years. This has been termed colony collapse disorder, which is very specific. It refers to the presence of the brood. So the developing larvae are still present inside the colony, honey and pollen resources. So there's sufficient food present inside the colony. And the queen is still present inside the colony, but all of the workers have absconded. They're gone. There's no dead pile of bees outside the colony. It's just really not clear where all of the bees went, right? However, if we start looking back at more historic patterns of beekeeping, as much as we can, since record keeping wasn't the greatest, obviously 150 years ago, or they may not have survived, it's becoming more clear that colony collapse may actually be more of a cyclical phenomenon. And certainly in the past 50 years, especially for looking at North America specifically, when we see these major declines within the beekeeping industry on a broader national scale, it can really be attributed to two causes. And the first of these is foul brood diseases, which were particularly bad in the 50s and then again in the 80s. And then of course we had the introduction of the varroa mite into the United States and North America, which actually involved a host switching event. So it's a native parasite of Apis cerana, or the Asian honeybee, and then switched over to Apis mellifera, or the European honeybee, which is the variety that is most broadly cultivated worldwide. However, we can call this cyclical, but the problem is that, especially since 2006, beekeepers are facing 30 to 90% of colony losses annually. This usually happens over winter when the majority of bees are not able to survive this period of time where their biology does shut down a little bit, even in more warmer areas that aren't necessarily as temperate. And so this is the issue, is that it becomes not sustainable from the producer's perspective. So, I mentioned two of the factors. We've got American foul brood diseases and then also varroa mites. But we already know, some people are, kind of, what causes colony collapse? Like we know that, you hear sometimes that we don't know what's causing colony collapse. We do, we know exactly what's causing colony collapse. We've got 6 main factors that are stressing our bees worldwide. The first of these is pathogens. There are a number of pathogens that affect honeybees. These can be bacterial, they can be fungal, and then they can also be viral. We've got parasites. The varroa mite is the worst parasite of honeybees. We also have an emerging mite. It's called Tropolaelaps. It is also a parasite of Apis cerana and has not been detected in North America, although it is kind of on the horizon as something that could potentially be introduced and be even more detrimental than the varroa mites. However, at this point in time, it doesn't seem like there's any imminent threat of this introduction, although it is something monitored for currently in the United States. We've got pesticides, which I think, you've got the perception versus the reality. The perception is that pesticides are having a terrible widespread effect on honeybees. However, it's very regional. It really depends where you are in the country, whether or not pesticides are playing a larger role in bee declines. Certain agricultural practices that are creating monoculture situations where you've removed so many valuable forage resources and put something in the environment that bees can't forage on. So for example, corn. The northern Midwestern United States historically has been incredibly important for honey production, but it's now almost wall-to-wall corn. There's also soybeans. You can get honey from soybeans, but it's not a particularly great resource for honeybees. So some of these ag practices go hand in hand with declining forage. However, declining forage can also occur in urban areas as well. So instead of establishing corridors in neighborhoods where everyone's planting in their home gardens things that are going to be valuable resources for bees, they're putting in things that look pretty, but they may be highly bred to be showy, but no longer provide pollen and nectar resources for honeybees. And then finally, we do have certain beekeeping practices. There are different paradigms of keeping bees, and some of them are actually harmful to the long-term survival of honeybees within a particular area. So as I mentioned, these two threats to Hawaii's honeybees specifically that I will be focusing on today, that I think are the most important for, for a broad audience of stakeholders to be aware of, are foul brood diseases, which are, this includes American foul brood and European foul brood. They're caused by two different agents, microbial agents, that they're two separate diseases. They are also present on all of the islands currently. So these were, or American foul brood, at least we know, was introduced back in the 1920s into the state, and it has since spread to all of the islands. And then varroa mites, which were introduced back in 2007. Currently, we only have Oahu and the Big Island that are positive for varroa mites, with Kauai and Maui counties both being negative. And the idea is to keep it that way. But the first step towards making sure that, and this is something whenever I'm meeting with beekeeper groups, we need to really have very candid discussions on monitoring and making sure that, however someone chooses to manage their bees, whether they want to be organic, they want to go more holistic, or if they're a conventional beekeeper, conventional practices, it's really important that they are monitoring each one of their colonies consistently. And I always encourage people, keep a log. Every time you go into a colony, if it's healthy, write that down. What are the things that you smell? Sometimes smell can be the first indicator that something bad is happening inside of your colony. So, what you're looking for upon entering an apiary are excessive amounts of dead bees or debris at the base of the colony. Maybe this is going to be indicative of a fungal disease, chalk brood. We see that commonly in Hawaii, particularly in the wetter areas. It's also not something to be particularly concerned about. You're always going to have background levels of chalk brood. Chalk brood is not going to kill a colony. You're looking for things like fecal spotting and diarrhea on the outside of the colony. If it's on the inside, you really have a problem. But certainly in Hawaii, we'll see it on the outside. And this is indicative of nosema disease, right? This is a microsporidian single-celled parasite in the gut that causes diarrhea. I'm looking for adult behavioral differences and obvious signs of disease. So in this case, we have an adult bee in the upper right corner that has shriveled wings. That's because it's positive and has a high load of the virus called deformed wing virus. This bee is going to die young and she'll never be able to fly. So she's not going to be able to contribute to many of the tasks that are required to keep a colony functioning. Finally, looking for things like a spotty brood pattern. And abnormal cappings, any type of weird smell. Now, spotty brood in and of itself does not, you find this almost all the time when you go inside of a colony. It doesn't mean that something bad is happening, but it's important to take note of it whenever you do see it, in case it can be an additional symptom among others that you're observing. So as I mentioned, we have a number of different viruses. Deformed wing virus is the one that is the easiest to detect because you can see the shriveled wings. Sometimes it's one wing, maybe both. A lot of times bees will be positive for the virus, but be asymptomatic, and so not show any shriveled wing phenology. You can also see acute bee paralysis virus, Israeli acute paralysis virus, and both of these viruses can sometimes manifest as bees being hairless inside the colony. They'll be black and kind of shiny. So sometimes seeing this symptom can be confused with German bees. So we do have German genetics here, mostly on the outer islands. So like Kauai and Maui, you'll see more of that. And the bees themselves are just darker, so that could be confused, but these are bees that are shiny and they have no hair. They're not supposed to be as black as they are. So that can be indicative of these two viruses. But by and large, most viruses, bees can have high or low loads of them, and they'll, you can't tell just by looking at them. I mentioned chalk brood and then nosema disease, which are both fungal. And then finally, our two most important bacterial diseases, American and European foul brood. Now, monitoring is important, and beekeeper education is so important because in Hawaii, we do not have in-depth diagnostic screening services. So beekeeper knowledge and education is really the primary tool to diagnosing and then intervening as soon as possible once a threat is detected. So I'm going to shift now and talk a little bit more in depth about the two foul brood diseases. So the first of these is European foul brood. It's caused by the bacterium Melissococcus plutonius. I mentioned smell being important. When a colony is sick, it's going to smell sour. That's not the bacterium causing this, but there's a number of co-infections associated with this disease, and that's what's going to cause this sour smell. This is becoming increasingly common on the mainland, according to the Apiary Inspectors of America, although we're not really sure why that would be the case. It's also becoming more difficult to treat. It's spread by nurse bees who are feeding the larvae, and then they're moving these bacterial spores around inside of the colony. And what happens is the bacteria will actually compete for food in the gut of the larva, and so it begs for more food. And when this happens, it actually causes the larva to shift from this like classic C-shaped pattern. So this is going to be a healthy larva inside of a diseased colony. They should be C-shaped. The reason being their spiracles, or the way they're able to breathe, is located on only one side of their body. And as food's placed in there, they just kind of move in a circle the entire time, eating the food in a circle. But if they're starving, they're going to deviate from that and kind of put their head up, begging for more food. Now, the nurse bees are onto this. If a larva is begging for food, something's not right, and they'll actually reject it and then remove it from the colony. But it'll take on this twisted appearance, and then as it starts to die, the trachea will become visible, and it'll turn like a yellowish color and then a brownish color. The larva will then liquefy, and then it's eventually going to form a rubbery scale at the bottom of the cell. Now, that's not going to be on the inside of the cell, but actually on the bottom, right? So these are sitting inside a colony vertically. So it's going to be kind of like right here on the bottom. That's where you're going to see these scales forming. So the good news about European foul brood is that, while it's a really bad disease you don't want, if it's an otherwise healthy colony and this somehow gets introduced into the system, as long as you have a sufficient number of nurse bees inside of the colony, they can overcome a mild infection with a strong nectar flow. I recently responded to an outbreak of foul brood on Molokai. And the colonies in the apiary that was particularly heavily affected, there was no nectar inside of the hives. So there was clearly, for whatever reason, those bees were not able to access the nectar flow, whether they had the queen died, they had to replace the queen. And so all of a sudden you've got this gap as far as the number of bees that engaged in foraging behavior. So maybe there was a nectar flow, but they weren't able to take advantage of it. So in that case, the recommendation would be provide them sugar water as a nectar flow supplement. If you're dealing with more moderate to severe infections, it's good to requeen. And what this will do is disrupt the egg laying cycle. And then allow the nurse bees time to go in and remove all of the diseased larvae, and that'll help the colony kind of reset. You can also shake the bees onto fresh frames and then feed them sugar water, but then destroy all of the old frames and all of the old larvae. If you want to treat with antibiotics, this is an option, but they have to be prescribed by a veterinarian. American foul brood is actually much worse than European foul brood. This is caused by a different bacterium, Penniid bacillus larvae, and this actually causes a really strong decaying smell inside of the colony. This is by far the most serious honeybee disease and has been responsible for numerous collapses since, as I mentioned, on the mainland, the 1950s, 1980s. The spores can persist for decades, and this is the major issue with this. So we know it's been confirmed up to 80 years on old beekeeping equipment. They're still finding viable American foul brood spores. This is again going to be spread by the nurse bees as they're feeding the larvae and they're moving the spores around inside of the colony. And then it can also be spread between colonies by robbing bees, honeybees are very opportunistic. If they sense this colony is weak, yes, well, let's go in. I'm sure they have a bunch of honey and pollen in there, so they'll just go and take that and bring it back to their colony. Well, unfortunately, they're also going to bring back any diseases that are present inside of that colony and potentially contaminate or infect a healthy one. And then also through contaminated equipment. So oftentimes, a colony will die, and instead of diagnosing why that colony died, a beekeeper will just recycle that beekeeping equipment. And now recycling the equipment is not necessarily a negative thing in and of itself. The problem is if that colony died as a result of foul brood and you didn't get in there until a little bit later. So maybe you had small hive beetles or wax moths that came in and then ate all of the evidence of this disease. You're going to assume that maybe it's okay and then just reuse the equipment. And that's going to be a problem because you're then going to infect the next colony of bees that gets put in there. As far as visual diagnostics, the cells inside the colony, you notice these are not, a healthy looking kind of creamy brown color. They look a little waxy, they're sunken. They've got some punctures here. That's from the gases escaping as the larva decomposes. And you're also going to have a positive ropiness test. So if you take a small stick and you see a gross brown goo inside of a cell and pull it, that is actually, if it's American foul brood disease, it's the only disease that will cause this to happen. You can pull it out almost 2 inches. If that is positive, you 100% have American foul brood inside of your colony. Treatment for this disease is unfortunately, it has to be drastic. If a colony is positive, regardless of the severity, it has to be burned. If all you see is one positive larva, that means the entire colony is positive. So the frames, the hive body, and the bees all have to be burned. So acquire a burn permit, dig a hole in that location, wait for night, close up the colony so all the bees, so all your foragers are back inside the colony, and you have to burn it. And you should never harvest the honey because this causes the spores to then potentially contaminate harvesting equipment. Some people like to advocate for the shook swarm method, where you just take all of your adult bees, shake them into fresh beekeeping equipment, and then requeen, and they think that that'll be okay. But what's likely going to happen is you're going to spread spores to new equipment. If this is attempted, the adult bees have to be treated with an antibiotic. If you have asymptomatic colonies in an apiary, these should also be treated for antibiotics, and then they need to be monitored for infection very closely, because again, if they become positive, they need to be burned. So foul brood is both an old and an emerging problem. So it was really common in Hawaii through the 1950s. It was actually, at one point in time, Molokai was the world's largest producer of honey, and American foul brood decimated the beekeeping industry in the state. And so many people don't know that. It never truly recovered after this happened. Since 2012, we've had at least 11 confirmed cases, but we have really spotty record keeping in the state. Most of these were included on or occurred on the Big Island. This most recent event that I responded to, we actually had a co-infection, which made it really difficult to figure out what is this, what are we seeing? Well, it's because they had both European and American foul brood, so that really complicated the diagnosis. As far as European foul brood, the incidence is unknown, but it's assumed to be relatively rare in the state. One of the big problems and why this may be becoming a more emerging issue in the United States and in Hawaii is that most beekeepers are younger now, and they have really limited experience with foul brood, which they may underestimate the virulence of this disease, or it can be misdiagnosed because they simply don't know what to look for. And then more experienced beekeepers have also reported that, especially with European foul brood, now that it's becoming an emerging problem on the mainland, is that the symptoms are not the same that they remember from past decades when they would have experienced collapses as a result of this disease. So, beekeeper awareness is really important. So here's a map of the, incidences just since 2012. The majority of these cases were on the Big Island. I don't think that's to say there's more of it on the Big Island. I just think it's the Big Island where it was being reported. There's nothing, since 2012 reported from Kauai. That's not because it's not on Kauai. It's just because it hasn't necessarily been reported there. And then it's likely much more prevalent statewide, but it's being misdiagnosed as either a dead out or some type of a queen failure. And the beekeeper is just not getting there before wax moths can destroy all of the evidence. So I mentioned veterinary intervention. If for some reason the beekeeper wants to treat with antibiotics, and that's totally fine, there are 2 products, or 3 antibiotics that can be used. You do have to have a patient record with a veterinarian who's capable of prescribing these now. And so for individuals who may not have a veterinarian, or if you live, like I know in Molokai, they don't have a veterinarian present on that island. Janae Odani, Dr. Janae Odani is our associate veterinary specialist based out of UH Manoa, and she is available for telehealth consultations and can prescribe antibiotics that way if necessary. There's also 2 test kits that beekeepers can purchase online for EFB and AFB, which work very similar to a pregnancy test. So it's always a good idea just to have a couple of these on hand in your beekeeping kit. What I tell people is don't test an asymptomatic larva, test one that looks nasty. That's what you want to know, right? If it's asymptomatic, it doesn't matter. You want to know what has made that sick one very sick. So moving on to Varroa destructor mites. These are currently present, as I mentioned, on Oahu and the Big Island. These are an external parasite that feed on the fat body, which is the equivalent organ of the liver in insects. This feeding actually weakens their overall immunity. As I mentioned, their native host is Apis cerana, or the Asian honeybee. In Hawaii, we do have very strict state laws that ban the movement of used equipment and bees inter-island. And thus far, that has been sufficient to keeping this parasite restricted to just Oahu and the Big Island. So beekeepers on the outer islands, I know it's popped up on Maui a couple of times, but they were able to immediately eradicate it when that did happen. And as far as the development and reproduction of this mite, what happens is a foundress mite will be present on the adult honeybee worker inside the colony, and what she'll do is sense when a larva is about to undergo pupation. So she'll drop off of the adult and go into that cell. The cell will then be capped by the nurse bees, and then the larva will undergo pupation. So this foundress female is going to lay an egg. That egg is then going to hatch. If more than one egg is laid inside the cell, she'll lay a male and a female egg, and so the siblings will mate with each other and continue feeding on the pupa. And then when that pupa emerges as an adult, the mites can then disperse and infect other individuals inside the colony. Now, on average, there's 1 mite produced per worker and 2 per drone, which are the males, and the males are quite a bit larger, so more mites can be produced per drone cell. In Hawaii, because we don't have a true winter, which disrupts egg laying of the queen, we have larvae being produced year round. And it means that in a single colony, you can have an 800-fold increase within a single year. So if you're not treating for these mites, you're gonna have a lot of mites. And then again, they can move between colonies during robbing and drifting events. We do have 9 chemical treatment options that are registered for use in Hawaii. 6 of these are organic, and then 3 of them are conventional. It doesn't matter which one you choose to use, they're all effective, but always follow the label recommendations because some of them cannot be used if you have honey supers. Some are going to be particularly detrimental to larval stages. And so it's important to understand the biology of your colony at that point in time when you're treating, and make sure that you're treating appropriately. Now, the Apiary Inspectors of America recommend treating when Varroa mite levels exceed 3% inside of a colony. They're actually talking about lowering that to 1% on the US mainland. For whatever reason here in Hawaii, and the research has not been done to explain why, you can probably let it go a little higher to maybe 5% before you need to have a chemical intervention. A drone brood removal is a non-chemical mechanical means of removing the larvae or the mites. So as I mentioned, the drones can have more mites developing inside of them. So if you go through and remove them, you could potentially be lowering your mite burden overall. And then the use of screened bottom boards. So this is going to be most effective if you know you have a hygienic strain of honeybees, because they're going to groom and chase off the Varroa mites. If you put a little oil in this pan, then when the mites fall, they'll get trapped in the oil. So the reason why controlling the mites is so important is because, as I mentioned, they lower immunity. They also transmit viruses. And the worst of these is deformed wing virus. Because it was relatively recent introduction to Hawaii, this group was able to come to the state and be able to monitor how deformed wing virus diversity changed after the mite had been introduced onto various islands. So if you look here, I know we're kind of constrained for time now, and I don't want to spend too much time talking about this, but effectively what this graph is showing is that deformed wing virus is less common on Varroa-free islands. It's more diverse on Varroa-free islands, right? So that's good. The issue is that when Varroa is introduced, it increases the prevalence of deformed wing virus in colonies and transmits more virus to the bees. And we also see that there's a reduction in virus diversity such that you have fixation on one very virulent strain of deformed wing virus. So that's what's going on. So this is my last slide as far as barriers to preventing the spread in Hawaii. One thing is we have no statewide beekeeping laws. The ordinances are actually on a county by county basis, and they're kind of piecemeal, although the Big Island is working to rectify that, at least locally. Honolulu County has a relatively good model that Big Island is going after, but Maui and Kauai don't have anything. So this is really confusing for individuals looking to take up beekeeping as far as, what's allowed here in Hawaii versus what's allowed on the mainland. Statewide, we have inactive or non-existent county beekeeping organizations. So at the local level, there just hasn't been organization, with the one notable exception being the Big Island Beekeepers Association. In response to the foul brood outbreak on Molokai, the beekeepers there are now reinvigorating their group. So I was really happy to see that. But the rest of the state just doesn't have a whole lot. I know there's also one on Kauai, but my understanding is that they haven't been meeting as frequently. This means that we now have an education void that's being filled by self-taught hobbyists statewide. These people are spreading information on the broader role of honeybees in Hawaii's ecosystems. Let's be clear, they are an invasive species. They are a very successful invasive species in Hawaii. The native systems here did not evolve with honeybees. And actually, a lot of our cropping systems do not themselves require honeybee pollination, with coffee and mac nuts being the two notable exceptions. And this is unfortunately promoting really ill-informed beekeeping strategies that in many cases undervalue the importance of treating for varroa mites, and also what to do whenever a colony is weakened by disease. We also unfortunately see complacency, which is, I think, in many cases resulting from a lack of education on varroa negative islands. And so I really would like to see a program, which I'm working on, where we're going to go into all these varroa negative islands and really educate them. Like, this is how you monitor for your mites. And even though you don't have it, you should be monitoring monthly in all of your colonies to make sure that it's not showing up, so that eradication can happen as soon as it does. And then going into, again, areas where it's varroa positive, and making sure that beekeepers have this best available information on how to treat for varroa when we know that they do in fact have it. Okay.
Thank you, Chrissy. So we have question and answer. Maybe we would use 5 minutes of break for the question and answer also. So I have a question for Anna. So Anna,
Yes.
So the stink bugs have some kind of gut symbiont, yeah?
Mm-hmm.
So is there any research on bacterial symbiont for BSMB?
Sorry, is your question on whether or not research?
My first question is whether or not the BSMB has a bacterial symbiont in the gut.
So they actually they have two, yes, they have some natural symbionts in the gut, just part of their microflora, just like we do, right? That helps them digest. There's some research that has been done to kind of characterize that, but not for control. Mm-hmm. However, they also have a Nosema virus, Nosema maddoxi, I believe, that is in a different part of their body, and that causes major problems with when we try to maintain them in colony. They don't molt very successfully, and we've surveyed. There's been work led by Ann Hayek at Cornell, surveying populations of BMSB throughout the country for Nosema populations. And they are prevalent at low levels. I think it's about 10% in most areas. So we are considering, we actually have an application pending. We're trying to use culture and apply Nosema as a management strategy is one of the research objectives, but we'll see.
Okay, in connection to this question, I have another question.
Okay.
So there is an adult female will lay egg.
Yes.
And the eggs will hatch. No, adult female will lay egg and the female will smear bacteria on the egg, right? Mm-hmm. And when the nymphs will hatch, they will feed those bacteria.
Yes.
So they will be, the nymphs will be now have a bacterial symbiont. So is there any potential of doing research to eliminate bacterial symbiont in the BSMB? So that the bacterial symbiont is basically for digestion and maybe reproduction, or maybe for some kind of adaptation. Yeah, is there some...
Right? So when the female oviposits, she is coating the eggs, and the first instars I mentioned, they're not crop pests because they're feeding only on the egg and they're picking up those endosymbionts which they need. If we sterilize those eggs, if we bleached or exposed to really, really high temperatures that kill that endosymbiont, which is external at that point, that they don't survive very well. So there's, yes, it exists. No, it's not a current, at least I don't think anybody is working on using that as a management strategy right now.
So we can look through the chat box, and there is another question. Because we don't have real winter, so we don't have a winter season here in Hawaii. So would it be possible if BSMB is in Hawaii, would there be possibility of more than 2 generations per year?
Yes, it's very possible. So if there's a report, there's a paper from 1930 in a tropical area of China where they suggest they have 5 generations per year. However, nobody's been able to really collect BMSB from that area of China. So what we think is happening more, because they have these overlapping generations, is that they have multiple population generations there, but probably not 5. So if you don't have a true winter, what we're seeing in there's some evidence in Florida and parts of California where they don't go into reproductive diapause. And so, but they will slow down a little bit during that time period. We don't know. Those populations are not very large. We've still only seen about 2 generations per year, but we don't know yet. I would not expect them to go into a full diapause where they're overwintering in a non-feeding, non-egg laying state. But there may be a period where they're not as active, or we could just extend the life cycle a little bit. So you may have maybe 3 generations per year. That has been modeled under some climate change scenarios for 3 generations per year. But once you take away that photo period requirement, I think 3 is about the maximum that's been modeled so far.
So the diapause state is the adult female? Or the egg
males and females, yes. So when they're diaposing here, they are not feeding. They're reproductively immature. They've never mated. They've never laid eggs.
So Anna, you talked about a little bit about the host plants that might be host of BSMB. So some of us asked, is lychee would be the other host?
That's
plant?
A good question. So I looked at the top 10 or 20 agricultural products for Hawaii when I came up with that list, and lychee was not on that list, or maybe it was an outdated list. I apologize if it was. I don't know. I haven't seen anything in the literature about lychee, so I can't answer that. But it's possible. I mean, it grows on. It's a tree fruit, correct?
Yes.
What family is it in?
Lychee? Sapotes?
Yeah.
I don't know.
We don't have it here.
Yeah.
I mean,
We have lychee and rambutan and lots of other tropical fruits.
I mean,
yeah.
I think a lot of the they really like rosacea plants, but and legumes. So, but as long as there's not really strong host defenses and it times up with their phenology, they don't seem to be that particular.
So they are polyphagous. Yeah, they can go anywhere.
Highly.
So yeah. So corn, soybean, whatever. Vegetables as well. Yeah.
Thank you, Chrissy. You say
like eggplants and tomatoes, maybe.
They'll feed on eggplant.
Yes. So I have another question. You talked about trap cropping, right? Perimeter trap cropping. Oh no.
Yes.
Yes. So we spray aggregate pheromone on perimeter trap cropping, or just a border crop, but with the same like a pear or pitch.
So when we did trap crops, it was part of our organic research project. So we were using a mix of sorghum and sunflower on the perimeter as kind of to block the idea was to provide a more attractive host plant than the peppers, which we were using as a cash crop. We were not applying insecticide. We were not using it. We did try it with an aggregation pheromone and it didn't do anything. We see about a 2-week delay. Essentially, when we use the trap crop that's untreated, we see a 2-week delay between the time they go into the cash crop. So if you're able to treat that trap crop, then you potentially could provide some management options. And thank you for those of you who are providing information about lychee. That's really interesting.
Okay, so I think the next question is for Christina. Christina, are you saying corn, soybean reduce bee population because the bees don't really like this crop? Can you explain if I got that wrong?
So the question is like, honeybees don't like corn?
So are you saying corn and soybean reduce bee population? So if you have corn and soybean, it will reduce population.
Right. Okay. So I mean, okay, yes and no, right? So if you have, I mean, if you're taught, it's not like, oh, we've got someone planted corn on Kauai. Okay, no, that's not going to reduce bees on Kauai simply because corn is present on Kauai, right? So what I'm talking, when I referenced that, I was referring to the time that I spent researching in South Dakota, where you have, I mean, there are no longer trees, there are no longer weeds on the side of the road. It is simply one species. So just corn being present in the landscape is not going to kill the bees. And actually, they will take advantage of and collect corn pollen, even though it's not like the best resource. But if it's the only thing available, they will in fact collect that and feed that to their developing babies. And even though it's not, it's a wind pollinated crop, like the honeybees will still take advantage of that. I know you can get honey from soybeans. I think it's a really dark honey. My understanding is correct. So there are resources available from these two, in these two monoculture systems. So the plants themselves are not going to kill the bees, right? It's going to be kind of more of a secondary effect where you no longer have plant diversity, which means you don't have a diversity in nutrition that's available. And so, conservation physiology, plant diversity as a means for getting nutritional diversity, that's my area of like research interest. And so that's what you're actually losing. So, you may have suppressed health of the bees. Now Varroa comes in, now some viruses come in, right? So it's going to be a more of a cascade that's going to contribute to declines. But I mean, there's plenty of honey production and bees that are present in the upper Midwest. I mean, the largest beekeeper in the world is actually up there. He's got around like 150,000 colonies. So, I mean, he's still able to make it work. So no, that's not, the corn and soybeans in and of themselves are not going to lead to drastic reductions in bees within a particular location. It's going to cause, additional stressors that with everything else of the 6 that we talked about, that's what's going to kind of have that detrimental effect.
Okay, thank you. So I have one question. So if a honeybee will forage on one crop or one insectary plant or whatever, can they, so my question, I don't know, can they forage on 2 different crops in the same day or same period of time?
So honeybees have what's referred to as a high forage fidelity. So if a single honeybee visits a macadamia nut flower, right? Or she goes to a mac nut tree or a trap crop, whatever that specific species is, so sun hemp, let's say. So she's visiting sun hemp, she's going to collect resources, either nectar or pollen, because it's whatever she's designated to do that day. She's going to then recruit to that resource. That single honeybee is only going to forage on that resource during that day. Now that being said, a strong colony at the peak of summer can have tens of thousands of individuals inside of it, and so you're going to have within a single colony foragers going to multiple different plants, but the individual bee is only going to visit one in a particular day.
Okay, so that's why we get only macadamia nut honey in the market, or something else honey in the market. Yeah.
No, you can. So you can get Kiave, like pure Kiave honey. You can get a relatively pure mac nut honey. And a lot of that also has to do with the fact that we think Hawaii, it's warm year round, there's food year round. There's not. We don't fully understand what bees forage on here. But macadamia nuts can actually, I mean, it's one of the only things that's blooming that time of year. And so that's why you can also get these really pure honeys right there, because it's, the honeybees need it, right?
What if there is a mixture of insectary plants?
So honeybees also, like the way that they're able to locate a resource is they're looking for a lot of one thing blooming in an area. So they're going to hone in on like one color or, one particular smell. Now that doesn't mean you can't have a mix of plants in an insectary planting. But, especially if you're dealing with a smaller scale, like a smaller farm, it may be better to just have one, if you really want to bring in honeybees to a location. So if you only have maybe like a 20-foot strip, just plant one species in that 20-foot strip. If you're dealing with a much larger, location, then having more diversity is fine. But the honeybee themselves, say you've got buckwheat I don't know, buckwheat, sun hemp, and sweet alyssum or something in your mix. You know, they're going to still find it, and they're still going to forage on all three of those. So,
thank you. So we talked about the pathogen causing disease, varroa mite, and what else? Is there any small hive beetle? What about small hive beetle?
Yeah, you did mention small hive beetle. So that is another pest that has been introduced and is now all over the state, unfortunately. It's a pretty bad one, but it's one of those things like if the colony is strong and they are healthy, they can keep the small hive beetles at bay, right? That's not going to be the issue. If you have a slime out, which is when the small hive beetle larva is the economically damaging stage, and it's their feces that actually has a bunch of yeast in it that'll ferment the honey. That causes them this, like, it just looks slimy and it's nasty and you can't harvest the honey. It's called a slime out. You know, if that happens, your colony was weak for another reason. The small hive beetle itself is not going, I mean, I've had beekeepers tell me that was the case, but then, you know, they're also not able to let me know, well, what was your varroa mite levels? Like, well, they don't know that. And so, I mean, in my opinion, my professional experience, it's more likely something else that weakened them. But the major thing that you saw, like the perception, was that it was the small hive beetle came in and you know, itself caused the colony to die, but it would have been something else first. Now, that being said, I think that you can, like, small hive beetles can also develop in certain fruits. They can complete their life cycle in certain fruits here in Hawaii. So there may be certain locations in the state where you've got, you know, wild fruit. I don't know if it's, was it strawberry guava? That could be the case. And that you just have a massive, like, emergence of the small hive beetles and then they're getting in there. So I suppose it's possible. But I think that it's less likely than a secondary cause of
okay, so there is lots of questions, and some of the questions are already been addressed. And let's see if we have more questions. Okay, so I would take a chance. Maybe one more question for Anna. So Anna, so there is a what do you call? The natural enemy, TJ, Trisolocus japonicus. It is accidentally introduced in the United States.
Yes.
So the percentage parasitism is very less, right?
Right. So in China, there's reports of what we call TJ, parasitizing about 67% of BMSB egg masses in mulberry and peach plantings. We're getting about 2%.
So,
There's
no.
some reports where it's higher, higher parasitism by a complex of parasitoids, but we're not at that level yet, and we're trying really hard to figure out what's going on.
So what is the percentage parasitism of TJ in its native range?
About 67%.
60-70. Mm-hmm. Okay, thank you. So thank you, Anna, and thank you, Christine. Dara is from University of California, Riverside. I cannot see. Surendra.
Can you see me?
Yes, now, yes.
Yeah.
So Surendra Dara, he's a cooperative extension advisor for entomology and biologicals. I don't know what is that, biological. So I think Surendra's expertise is in IPM, exploiting insect pathogens, if I'm not wrong. And he's very experienced. One time I asked this question, what is your experience? So he told me my first job is in Kenya. Then after that, there's lots of other jobs. And right now I'm in this job. It looks like he has around 30 to 35 years of experience working in insect pest management from India, Kenya, Canada, wherever, in all different parts of the world. And I think he got that doctoral degree from University of Virginia Tech in 1995. So with that, I would turn into Surendra, and Surendra is presenting on extension strategy for addressing invasive pests, the California experience. So Surendra.
Good morning or good afternoon, everybody. Thank you, Roshan, for inviting me to speak. Let me turn this thing off. So my presentation is going to be mainly about extension strategies, not a whole lot about the pest and other, how to control and so on. It is because, invasive pests are going to be there, like with the global trade and travel and everything else that's going on, we are continuing to have these issues, and how well we are prepared to address situations like this is very important. So I'm going to use Bagrada bug and the spotted lanternfly as two examples of my experience working with them, and see how it actually either helps to manage a pest that is already there or prepares us for a potential invasion. So if we want to look at the general strategies when we have an invasive pest that is in the country, but hasn't gotten to our area, the most important thing is to have some regulatory guidelines that prevent the potential invasion to the new area and also its spread, depending on how it is going to be transported on a plant material or inanimate objects and so on. Then the next one is the outreach that helps increase public awareness. We want people to be aware of this potential threat, or basically a lot of people don't even know about invasive pests unless you see something like this murder hornet and so on. A lot of people are not aware of what kind of potential damage they might run into, especially if it is not especially if it is only an agricultural pest. They don't know. So it is very important to increase the awareness for multiple reasons, to prevent and report if it is there, and also to control. The next one is engaging the growers or affected parties and public. This is again not just to let them know what is coming and what is there, but they can play a critical role in reporting a pest incidents, and they can help us identify early enough. Sometimes all it takes is an egg mass. So if someone is familiar with this potential pest or a pest that's coming to that area, then they can basically help us identify, destroy it. And also, if it is already there and spreading, they can help us map the distribution. So it is very important to have that kind of engagement. And the next one is area-wide effort. If there are any pesticide applications that need to be made or anything else, it is not about biological control yet, but this is initial stages. We could, use one of these approaches, area-wide management approaches. Then ultimately, we need to have research depending on the kind of pests. We could either have these arthropods or pathogens that could be brought and released for this classical biological control. And at the same time, as Anne mentioned, like with the brown marmorated stink bug, you see a variety of strategies, aggregation pheromones, or pesticides, mating disruption, and so on. There could be so many things that we could use as control option. And finally, we need to develop these IPM solutions and then take the message out. So these are typically various phases or strategies that we use for addressing an invasive pest. And then I'm going to start with this briefly introducing you. Some of you might have seen my new IPM model, where you have the central part is about the management aspect, and then outer two circles are the business aspect of pest management and the environmental aspect. I'm going to remove those two, and we will just focus on the four components of the management. Where you see the green one, everyone is familiar. We have a variety of control options. So we need to be aware of these. Even before the pest comes to our area, we need to understand, okay, if it comes, what kind of options do I have? What can I do? Which one is available to me? And then, and a very important thing is this knowledge and resources. Understanding the best pest biology is the most important. What kind of life stages there are, and which one is vulnerable to a particular tactic. Can we use mating disruption? Can we use behavioral control? Or is it more on the perimeter of the field? Or does it prefer alternative hosts or wild hosts more than the crop, and how do we manage and what kind of control options do we have are important. Then the planning and organization, it is more for the growers to manage it, not for extension and other parties there. But again, when it comes to the communication, we all need to communicate, like presentations like this, and then various things like informing the public, informing everybody involved. So all these things help us to prepare for and manage these invasive pests. And the first one is, as I mentioned, this is Bagrada hilaris. It is from Africa, and it is a hemipteran pest that feeds on, it has a variety of hosts. It primarily feeds on cruciferous hosts, but it also has several other cultivated crops and wild hosts as well. So whenever you have a lot of these wild hosts, it is always a challenge, because we can only control weeds to some extent, and these wild mustards and wild brassica crops are all over in the hilly areas in many places. So when they dry out, and these insects, or any of these insects that feed on them, can move on to crop plants. So this is briefly, quickly, the various life stages of this pest. It and it is different from harlequin bug. The bottom right this is what you see. This is harlequin bug, and then these are Bagrada bugs on top of it. So we want to inform people or whoever is reporting this to us, they need to be aware of these differences, and sometimes it can be very confusing, and they might give you incorrect information about citing by an invasive pest. And these are various crops that the Bagrada bug can feed on, and depending on the life stage and then number of Bagrada bugs that are coming in, it can either completely, I will show you in the next slide. So, you can see a lot of them coming. And if it is a newly transplanted crop, then it can completely destroy that. And it can form multiple heads, or it can just do damage to the foliage or any other part it can feed on, and weaken the plant. So it is important to know where it is. Like, is the transplants or newly planted crop more vulnerable than the mature one, or how to detect the pest and what can we do about different life stages is all very important. And this has multiple generations, and it is not clear how they come. Like all of a sudden, even when there aren't many wild hosts nearby, they just come in large numbers. So the thought is that they can overwinter in plant debris and in undisturbed areas, uncultivated areas, and then they move to crop plants. And and I just want to show when it was first detected in California, and then how it spread to various counties within afterwards. And then, after 2015, I stopped monitoring because, we actually don't have this pest as a big problem now. So I just want to highlight something here. See, you see this, the first map, it was first seen in Los Angeles County, and in June 2008, I joined UC Cooperative Extension about 7 months after, and I didn't come across the news about this pest right away. After a few months or so, when I realized that my colleagues in this county, Imperial County, are working on it, because that it has spread there and causing damage to the crops, especially to organic crops, I started collaborating with them by suggesting some non-chemical alternatives. Here, it is important for us, like it is not there, not in our area yet, but looking at the pest biology, it is, it feeds on these crucifers and other vegetable crops. And a lot of backyard gardens, community gardens do have these crops. So in a farm situation, especially in a conventional farm, you can spray a lot of a variety of chemicals and control them. But what about organic crops? Especially this was a big problem in organic cole crops. Because you need good control options. So same thing could happen in backyards where chemicals may or may not be sprayed. And when you have these numerous households as sources of infestation, they can very easily spread to cultivated areas. So we need to be very proactive in detecting this, anticipating potential risk, and do what is necessary. But what happened was during my performance evaluation that year, I had a bad remark for working on a pest that was not in my counties yet. So I had to stop and then restart again after several months, almost after 2 years. So quickly want to show what I was doing on about this pest, like mostly writing these articles to alert people and growers about what is there and what needs to be done. And if you see, this is 2011 was when I wrote my first article, and it generated a lot of interest. And then almost 20 months after I wrote, that's when we had it in our county, Santa Barbara County, but I had to stop for nearly 20 months. But once it came, there was so much pressure, people calling and asking information, what do we do? What is this pest? And so I had to write several extension articles just to provide information about what it is, what can be done. And then, and also this is like various trade journals, and everywhere we have to use every possible resource to spread the information and, basically give them the information about identification and management options. And then I also put together this short, less than 3-minute video because so many requests were asking the same information. And, sometimes when you send them to these articles, they don't have enough interest to read the entire thing. So I just put this together for those who don't have enough time. So all we could do when we have such pest invasions is that to provide as much information as possible using all communication channels. And then once I started getting these requests, like I was responding, responding to the calls or emails and so on. Then another thing is they all ask, what can we do? But I didn't have any chance to do research, especially this is mainly for organic growers and those who cannot use chemicals. So I would suggest what I could based on my experience with other pests. And then, it is a hemipteran, we can kind of guess what to recommend. And then I asked them to provide their feedback after a week or 2 weeks, tell me what happened, like how were you able to control with these strategies? And then depending on that information, I could suggest, different strategies or the same strategies to other people. And then, giving extension talks, and then coming to research, it is like I did a lot of assays with entomopathogenic fungi and botanical and other insecticides. And then I also tried to do some field studies. And then also when I went to India, I visited some fields actually exploring for potential biocontrol agents. So these are typically what anybody could do: provide information, try to do certain studies, and develop classical biological control as the main option, and then provide other control options. So after a few years, this problem is no longer a serious thing in California. We have very few isolated fields in some areas. We don't have any issue on the Central Coast. And in the Central Valley and some other places, they do have some infestations again, especially in organic brassicas. So this problem kind of disappeared or under control without a whole lot of effort. So this may or may not happen with all kinds of pests. Then the next example is this spotted lanternfly, which is again another hemipteran that can feed on a very big host range. We have a lot of hosts, like cultivated crops and landscape trees and ornamentals and grapes and other ones like that. The tree of heaven is its most favorite host. It is also a weed in the United States, and especially we have California. This tree of heaven is distributed all over in almost everywhere in California. And then, grape industry and other stone fruit and other crops are at risk if this comes to California. So because of my interest in invasive pests, I started working on this, and I know once or if it ever comes to our area, then it is going to be a big problem. So I wanted to alert my growers and everybody basically, so that we can contribute to its, potentially prevent its invasion if possible, or immediately handle it if it ends up coming here. So quickly, I want to show its biology. It female deposits its eggs and covers with this waxy protective coating, and spotted lanternfly overwinters as eggs, and this waxy coating is very, very sturdy, and then it can provide excellent protection against cold winter conditions. And as you know, this is mostly in the East Coast right now, and we don't have in other areas yet. Then it has these three nymphal instars that are black with the white markings, and then the fourth instar has this red markings as well. And here we have this male and female, and then the life cycle continues. So this one has only one generation right now, but if it comes to warmer areas like California, what do we do? Like, does it have multiple or overlapping generations? It is quite possible because it is all about the environment and the speed of growth. So what it does is it feeds, it inserts its stylets, and then the infestation occurs in large numbers. You can see in the middle photo, like there are hundreds of these killed lanternflies, spotted lanternfly adults. So they feed on them, reduce the vigor of the plant, and then under severe infestations, they could kill the plant. And this vineyard was just sprayed 3 days before I took the photos. This was from Pennsylvania, and you can also see sooty mold growing on this from the honeydew secreted by these flies. So it is important to control them to protect this grape industry and peaches and various other crops. And it can also be a big problem in the urban landscapes or backyards where those host plants are. It can occur in hundreds and thousands, like the brown marmorated stink bug. So how does this pest spread? These adults deposit eggs on anywhere, not necessarily on the plant materials, but they can deposit eggs on these like boxes or vehicles or rocks and so on. So it could easily be distributed all over the United States from those infested areas, either these shipping materials, these boxes and so on. And there was actually an example of this, and I don't want to mention, but there was a shipping facility near to very close to this infested area where we visited. So things like that could happen where they inadvertently ship the material. And similarly, it looks like the egg mass looks like a splash of mud, so you may not even realize that it is related to an invasive pest. So it could come and spread to the new areas through multiple routes. So this is why it is important for us to involve public, inform everybody, so that they can identify and probably scrape these egg masses or report and contribute to the prevention of its invasion. And then other outreach and research, like write a lot of articles. Again, I started writing, and then especially this scientific article in Journal of IPM has been very popular. And then based on that, eventually people ask you to write or give talks, and you generate more extension material. And then one thing I am currently working on, I got funding from California Department of Food and Agriculture, was to do this outreach activity and map risk areas in California. So we recently started, and this is a preliminary map that my postdoc is working on. So what you see is like a very low risk to critical area, high risk and critical area, and the bottom left. So based on the percentage of its cultivated hosts, we rank different counties with the different levels of risk. So you can see, which one is less risky. I mean, which one has the less risk if it invades, and which ones have moderate and high risk. Although some areas may not have enough grapes, but the cultivated other crops are there, and they can also host this pest and become a big problem. And then this is based on uncultivated wild hosts. Tree of Heaven obviously is one of them, and based on, again, the percentage of wild hosts, this is the risk that we can anticipate. So these kinds of information is helpful to prepare. Preparation seems to be less important unless you have a problem, as we can see from COVID or any pest issue or anything else in our life. Until it comes to us, we don't realize how important it is to be prepared. So that's what we are trying to do with this pest. So, and then we also have a lot of IPM tools already. The first thing is to outreach, inform people, and then prevent and prepare people. And then once, or if it happens to be there, the pest comes to a new area, then we have a lot of strategies for different life stages, what we can do for eggs and nymphs and adults, and how we can also contribute to its IPM through removal of alternate hosts and so on. So this is what IPM kind of IPM we have, and basically prepare people. It is not to scare them, but prepare. The first part you saw, that alerting everyone that there is a potential invasion. Then here we are saying, like, yes, it could be there, but we have these strategies we can try to manage. And here I quickly want to give a general information, some kind of overview about these invasive pests. Like you're probably aware of all of this already. So when we have an invasive pest like spotted lanternfly in new area, it doesn't have all those controlling factors. Or like here it is a mutualist, is like a something that is beneficial to it, and these are their natural enemies and other competition. And if it has this enough host plants and the environment is ideal, that's when we have a pest problem. If the environment is not ideal, then like what we saw in the case of a Bagrado bug, it could be the environment, or it could be natural enemies adapting to this invasive pest and having those pathogens or predatory or parasitic organisms that might be controlling it, the environment or other things that kind of suppressed. Whereas, with the spotted lanternfly, we don't have that kind of control yet. So when we have an issue like that, all we have to do is, try our best to manage the pest with the knowledge we have from other pests or the ones we generate, and then quarantine. A very important thing, alerting everybody, even these kinds of invasive pest information should be there on all flights, and everybody should be aware of it. Only a few people or high risk areas are familiar with this, but we should have a focus on letting everybody, the entire country should be aware of invasive pests and their the potential of their damage. And then obviously we have to do research and then spread that information. These are typical things that we can do about any invasive pest. And then just a few final points, key points for addressing pests like this. So being proactive, very important. As I have been emphasizing, we don't have to be researchers. We don't have to be the biocontrol lab or a lab where it can actually work on these insects once they arrive, but we can be very proactive through extension, extension and other ways. And then we have to alert them, anticipating is one thing, and then working with the, in our case, we have ag commissioners, we have commodity boards like many places have, and then we can work with them and obviously our colleagues. We have to prepare everybody for potential invasion, and then, focus on this outreach. As I keep mentioning, outreach can be a tremendous component of managing pests, because that informs people, that provides information about how to handle a situation like this. And we can do this while these classical biocontrol and other control options are being developed. Then with the information we have either from its native country or based on the experience we have from managing other pests, we can put together guidelines. As soon as something arrives, we could have something already. We don't have to wait. We have, as you see, we have these pathogens, bacteria, viruses, entomopathogenic fungi. We have a lot of botanical insecticides, and then, obviously several categories of chemical pesticides. They can be used readily for, any of these things while the biocontrol options are being worked on. So this is the end of my talk, and if you have any questions, I'll be very happy to answer.
Yes, thank you, Surendra. So we will have a question and answer session after another talk. And thank
Sounds
you, thank
good.
you so
Thank
much.
you.
Yeah, there is a question in the chat box though, but we will address later. Okay, so next presentation is from Mike Melger. So he would be speaking on exotic pest pathogens of taro, their threat to Hawaii, and the challenge of developing reliable diagnostic. So Mike Melger. So Mike is the now maybe associate researcher in Department of Pests, and his expertise is in plant virology, and he also heads the UH Agro Security Lab. And he got his PhD degree from University of Hawaii, Department of Pests, around maybe 2013. And he's involved in lots of different projects, including CRB. And today, maybe his talk is mostly on maybe National Clean Plant Network project that he leads, and also National Plant Diagnostic Network with the exotic pests and diseases of taro. Mike.
All right, thank you guys. Can you guys hear me okay?
Yes.
All right, so
but this is a different format. Can you do the presentation mode?
Oh, I'm not in present. Oh, okay.
Yes.
Well, let me get
Still not. Still. Yeah. No.
You'd think I'd be good at this. I teach this all the time. Let's see now. Let's get going back here. That's interesting. Okay, let me get my Zoom up. Well, first, two apologies.
No problem.
First, for I'm missing the beginning of the class. Is this better?
Yes.
All right.
Yes,
First, yeah, I teach in the morning till 10 o'clock, so sorry for joining a little bit late. And the second apology is this talk is a work in progress, so it's going to be a little disjointed and a little hairy. So apologies in advance for that, but it's not a nice, clean story yet, but we're working towards developing one. But I still think there's some interesting updates here. So yeah, thank you for the introduction. My contact information is below, and I think I can keep this under around 20 minutes to keep us on time. So we work on a lot of crops, but taro is a personal favorite of mine, as we'll sort of see. Let's see here, let's get to this one. I don't think we need a lot of background on this, especially for the folks in Hawaii, but taro, Colocasia esculenta, sometimes depends on what taxonomist you ask to, they might call it antiquorum or variety antiquorum. Some people consider xanthomonas or xanthosoma or alocasia species taro as well, but they're a different genus. But it's an ancient starch staple that's been grown around the world, and here in Hawaii, it's grown both wet and dry. And all parts of the plants can be eaten, but only after thorough cooking. As anyone who's had undercooked poi or luau leaves knows, you need to fully cook this plant. Where is it being produced? Most of the product, even though the center of origin of this plant is probably the Indo-Pacific region, and some people go as far as Papua or New Guinea Island, Africa is dominating production, and that's where most of it's grown as a starch. Fairly modest production worldwide in 2019, about 10 million tons being produced. Hawaii, where taro is very important. Unfortunately, we've been in a decline in terms of production. The 2018 numbers is about 1,300 tons, down to from sort of a post-World War II high in the late 1940s of about almost 6,000 tons. So even though it's maybe not as economically or production-wise as important as it used to be, it still holds important cultural importance here in Hawaii. So what I want to talk about is what I call the trifecta of pests and diseases of taro. There's three of them that I consider the most major issues for taro. Taro leaf blight, caused by the oomycete or fungal-like pathogen called Phytophthora colocaceae, taro beetles, and the Alumibaboni complex. I'm going to focus on the last two though, just because we have taro leaf blight in Hawaii. So it's a threat, but we've been dealing with this threat for a while. What I want to focus on and what our lab tends to focus on are things that haven't established or just recently established. And so we always are trying to scan the horizon, so to speak, for some of the pests that we don't want to get here. And I think that's sort of been a theme of the previous talks and well, basically all invasive species talks. So I'm going to discuss a little bit about taro beetle and then a little bit about Alumibaboni disease. So taro beetles. Papuana species, they're coleopterans, scarab beetles, dynastids. There's, depending on who you ask, there's between 17 and 24 species in the genus. They probably native to the Indo-Pacific region. And again, a lot of people think probably they co-evolved with taro because most are present on Papua Island, which is again, or New Guinea Island, which is also thought to be the center of origin of taro. Here we can see a few of the different examples. We got these off of a nice scarab website. For a lot of people, they just look like CRB or or coconut rhinoceros beetle or CRB. Well, in the same, in the same taxa as them. They're just a bit smaller, probably about half the size. And similar to CRB, it's the adults that are the damaging life stage. So basically, they're the ones that are responsible for boring into the corms, producing these galleries, which allow secondary infection. The larvae themselves are fairly benign, if not beneficial. They're in they're feeding on organic matter and kind of in the soil and compost and so forth. So in that respect, they're very similar to coconut rhinoceros beetle. And basically they can just devastate a taro crop through their galleries. And again, so each of those galleries, for scale, that insect's maybe a centimeter to a centimeter and a half wide. So each of those boreholes are, probably about the size of a quarter. So they can go through a corm pretty well. And what they don't eat, secondary infection from things like Phytophthora or Pythium and other diseases or bacteria can gain entry. The impact depends on how well it's managed and where you are. Sometimes in Papua New Guinea, they're seeing losses as much as 80%. And that's typically when there's not much control efforts going on. We're not going to talk so much about the control efforts at this point. This is more of a preventative oriented presentation. In Solomon Islands, the other place where this is common and where a lot of taro is grown is a lot of farmers have just abandoned taro and switched to other crops or other starches, things like sweet potato and cassava. Even though those are potential hosts of the beetle, they're not the preferred host. This beetle will go to does have a wide host range, but taro appears to be the preference. So where are they? Fortunately, they're fairly localized. If you look at a few of them here on the CABI website, I'm not sure about the Korea thing. I see if you see it up there. It's sort of an undocumented thing, and CABI sometimes does that, but we can see Korea here. But this one's generally definitely through Oceania to some extent, basically coming down from Southeast Asia and through Fiji as well. If we look at another one, probably extends, so this one has been found in Kiribati, although I don't know if it's present in all the islands that are represented here. So we're inching a little closer to Hawaii. These are incursions from, several decades ago, but Hawaii is not too far away. And these things are apparently reasonably good hitchhikers as well. So they might follow some of the similar pathways that coconut rhinoceros beetle does. This one isn't in Fiji, but the other one is, and we all know Fiji is a fairly good transport hub for the Pacific, and plenty of direct flights to Hawaii. So what are some of the challenges with this? Well, right now, I'd love to tell you more about these beetles. And there's a couple of reasons I can't tell you more. One is, well, what they're finding is that the taxonomic keys that were established several decades ago tend to be difficult to follow, just because the beetle has, there's probably more variability within species than originally described by some of the people who published the original keys, some back from the 50s all the way through the 80s. And this has actually led to several misidentifications, and we all know that's always concerning when it comes to invasives. So there was always a lot of debate about some of the ones that made it to Fiji, what species it was, because they were having difficulty trying to follow the key and identify the species. And trying to walk back a previously thought identification is sometimes difficult, especially now because it gets out in the literature and it's hard to retract that. The other thing that's an issue is genetic data is largely non-existent for these insects, or for this genus in general. In fact, if you go to GenBank, where most of the genetic data in the world is housed, and you as they suggest, click on the organism name to get more information, you're going to be woefully disappointed. For this whole genus, basically the only sequence in there is a 16S gene for Woodlarkiana. There's no other genetic data available in there. So you couldn't even barcode these things if you were so inclined, whether you agree with barcoding or not, but they just try to make some identification based on genetics. You're kind of out of luck unless you've got your own database. So we've had several attempts to try and resolve this, but unfortunately COVID has had some of these, has been responsible. So some trips I had planned last year and again this year have been delayed to hit some of the islands with colleagues who are working on this beetle and start to try and develop some data. I'm by no means a morphological person or even an entomologist. I play one on TV, that's about it. But we can definitely help out with the genetic data and maybe try and gain some traction in developing some of the morphological work and maybe trying to work on the keys or identify people, or at least raise awareness about this insect. So at this point, I'd really like to talk about more about this, but that's unfortunately not so possible. But what I can talk about is, or at least we've made some progress in the last couple of years, is with this Alami babone thing, this unresolved virus complex, which I think is probably the most severe out of all of them. But fortunately, the reason that it's less known is that it's so isolated and it's almost so lethal that it wipes itself out. So it's basically the Ebola of taro diseases. It almost pitters itself out. So this is a poorly understood complex, and most people are supposing that it's involved. You need more than one virus in order to cause the disease, and some of the players that have been mentioned are the recently described Colocasia babone disease associated cytorhabdovirus, and then I'll skip one, and then Taro-vein chlorosis nucleorhabdovirus. So those are sort of two related viruses. They're both in the rabies virus family, but these are plant viruses. Then in the middle, we've got Pterobacilliform badenovirus. And then there's a closely related one that's, it's a different species from China, but it's just known as Pterobacilliform China virus. And then there's always the potential for more undescribed viruses, and that's highly likely in a vegetatively propagated crop like this. That doesn't go through seed to exclude a lot of these viral pathogens. So where is this? Well, right now it's only in 2 places. Well, really probably 3, but officially only 2, and that's Papua New Guinea and Solomon Islands. You can see them way on the east side of your map there. These are the only 2 locations where this disease has been identified. Off the record, Vanuatu has had a few suspect samples, and they get pulled and removed and destroyed, and there's no documentation of them. So there's an oral record of the disease being in Vanuatu, but there's no documentation of that. So what does this disease look like? This is relative, actually, I would consider rather moderate symptoms for it, but we do get severe plant stunting and distortion and chlorosis of the petioles. What I'm pointing at, what I'm interested here, again, somewhat, not too severe on one of the kiki coming up, but what I sort of wanted to point out, a symptom that sometimes shows up is this galling on the petioles, and that's a fairly distinctive, and you can see some of it over here, and that's going to come into play a little bit later. And some people associate this symptom with this disease, and it's a pretty rare symptom for taro, and a lot of plant viruses. Very few produce that sort of galling. So what's the impact of this? Well, Alami baboni is in some ways, it's difficult to describe. Alami is considered a disease that attacks the male varieties, which taro doesn't have male and female plants like, say, papaya does, but male is sort of considered the larger varieties. And so Alami, which is the disease that affects the large varieties, is generally lethal. The plants do not survive. So these larger, sometimes preferred varieties are harder to grow where the disease is present. Whereas babone, which seems to attack the smaller varieties, but it's probably the same disease, it's the same viruses, but just it's a varietal difference. That's my guess. I don't know if that's true or not, but that's my guess. It tends to be a little less lethal, and it's probably just those varieties tend to be a bit more tolerant of the virus. So in that case, in some of these smaller varieties, the disease is rarely lethal, and the plants will recover, and symptoms will reappear when they're stressed, but it's not as big of an issue. And again, when you read this about them being 2 different diseases, but then you just try and filter it from experience, and where some varieties are just highly susceptible, it's probably some of these larger varieties that are growing that are highly susceptible to this disease, and that wipes them out. And we've had cases of this in Hawaii as well, where we've lost varieties probably through the introduction of exotic viruses or other pathogens that were particularly virulent against particular varieties. So what have we been doing on this? Well, if you remember, just a refresher of some of the viruses that are involved, we sort of started to get involved. The one we could work on was Taro-vein chlorosis nucleorhabdovirus. So this virus, the reason we could work on this for a while is because it's in Hawaii. So we discovered it in a survey back in 2012, 2013. So it's been almost a decade now, but I'm sure it's been here much longer because it was fairly widespread. And this is, as the name suggests, causes chlorosis in the veins of the plant. The plant sometimes dies, sometimes it recovers, but it's probably the most severe virus we have of taro here in Hawaii. So we did a little bit of work on this, and a lot of frustrating work on this. One of the reasons was, just like with the taro beetle, there's only 1 genomic sequence available for this virus out of Fiji. And so when we were trying to do diagnostics, we were pulling our hair out because the existing primers that were developed by an Australian group didn't work for us. We would see symptoms, but they're always negative. So we worked on developing some new primers. This is really old data. This is from from almost 10 years ago. And so we have developed some. And if you look at this bottom chart here, oh, well, we lost it there. But in any case, the new primers tend to work a little bit better than the old primers, but they still weren't perfect. We were getting samples from other parts of the Pacific, and even our new primers weren't doing a great job. So to do this, we needed to get more information. So we traveled around and got some samples from other Pacific islands. And we sequenced their genomes. So Hawaii, Guam, Palau, Samoa, Vanuatu. We sequenced those genomes. We got some more information. Now we've got the whole genome to work with for developing PCR primers. And so where we are right now is we've got 5 that we kind of like, targeting either the replicase or L gene, the glycoprotein or G gene, and then a couple targeting the nucleocapsid or nucleoprotein gene, and the 3' and terminus. So these are probably the best primers we have for a molecular diagnostic assay. So we're gaining confidence that we can detect this virus in almost any sample now, but it's always a story of improvement. But we also wanted to tackle it from a serological point of view, basically an antibody-based assay. So one of the first things this is a lesson in don't try and do it the quick, fast way sometimes, or the quick, cheap way. So the first thing we did was synthesizing some peptide sequences from the nucleocapsid. We picked 3 hydrophilic regions hydrophilic regions from the nucleocapsid region, and had a company make some polyclonal antibodies in rabbits. Didn't work great for us. In fact, well, in some ways they worked too well. We could detect the virus in samples that did not have the virus. So the antibodies were actually a little too good. They were a little too loose, I guess, and a little too non-discerning. So our next approach was, a little bit more difficult and doing it, but we expressed the entire gene or the entire protein in bacteria, and then use that here to actually inject mice and develop some monoclonal antibodies. We had some issues with that. This might be a little too technical, but we had the expected size protein, but we were consistently getting more protein that was about half the size. So we think there might have actually been some cleavage here, and it might be from the bacteria itself. Sometimes when you try and express proteins in bacteria, they don't conform right, or they get cleaved, and we think that might have happened here. And we think these antibodies worked a little bit better, these monoclonals, but still not perfect, still not great. Still, we're still not satisfied with them. So our most recent thing is purifying the virus. And so we just did that earlier this year. And although it took a lot of work, this is sort of the gold standard way of developing antibodies. So we have purified, this is the bullet-shaped virus here, looked through a transmission electron microscope. So this is the best way for developing antibodies. So we'll be acting on these purified virions for developing antibodies, and hopefully we'll finally have a serological test after about 3 or 4 years. And that just sort of teaches you that sometimes the quick, easy way isn't the best way. And we should have just probably gone through the gold standard, which is producing or trying to purify the virions to begin with, which is probably the most technically challenging approach. So that's where we are with that virus. But what's let us do some more work on this disease, and so I know I've only got a couple minutes, so I'm going to try and wrap this up quick. We were able to get our hands on some legitimate samples. I know it's hard to believe, but these are all taro plants. So I would consider, especially the middle plant, a pretty severe infection of Alami bobone disease. And so we got several samples from Papua New Guinea. But 3 of them had this disease from 2 different villages. And so we started looking at this Colocasia bobone disease associated cytorhabdovirus. We found that one in all 3 of the affected plants. So there's probably a good chance that that's one of the causal agents. So we've been able to sequence the genomes of isolates that came from 2 of the villages. And it's probably running into the same story that we just had with taro vein chlorosis. There's a lot of genetic diversity in this virus, 82% identity between ours and the lone isolate that's been sequenced from Solomon Islands. And that means that PCR assays are going to be difficult because there's so much genetic diversity. So we're going to be pulling out our hair again, trying to develop diagnostics for this virus because it looks like it's going to have a lot of diversity in it. So just reiterating what I said there. The other one we looked at, Pterobacilliform badenovirus, that one was present in all of the symptomatic plants as well, or in one of them, I should say, but then a closely related one was present in all of them. But the problem is this virus is present in almost all taro plants, including here in Hawaii. So we don't know what it actually contributes to the disease. The one we talked about earlier, Taro-vein chlorosis virus, it was actually absent in these plants. So that questions whether it's involved or whether it's a replacement of the other rhabdovirus, the cytorhabdovirus. Maybe both of them are able to induce this when combined with something else. And then there's always the potential of other undescribed viruses. So one we actually just came out earlier this week on the 11th is a reovirus that we found in this. Well, it's been known that it's infecting taro, but finally we're able to sequence the genome of it and properly characterize and publish it. And the reason I bring this one up is because the most closely related virus infects rice, another monocot, and it produces galls in that plant. So one of the distinctive symptoms, it's kind of hard to see from this picture, but one of the distinctive symptoms is galling. And if you remember, that's one of the distinctive symptoms with Alami babone, or some forms of Alami babone, is this galling. So that's why we're interested in this one as being potentially part of the etiology of the virus, of the disease. And I'm not going to bore you with some of the other viruses that we found in there. Using the approach, we found a lot of new viruses undescribed in these plants. And so now the process is trying to work out, which is involved in the etiology of the disease, so we can develop diagnostics. So to summarize things, so I know that was quick, I was probably talking quicker than I should, but so yeah, to summarize this up is that the taro beetles and Alami babone, I believe they represent significant and I don't think there's any argument that they're understudied threats to Hawaii that warrant more study. Fortunately, one thing we've got going for us is if you look at H2A's rules, that we do have limits on bringing in taro. Noted here is Solomon Islands, and they're worried about Alami babone. It's always good to add Papua New Guinea to this as well, as we know it's there, and our samples demonstrate that as well. So at least we've got safeguards for permitting and quarantine for any propagative taro parts. So good on HDOA for having that. In terms of for Papuana, I think there's no question we need to improve morphological keys as well as genetic data, and that's going to happen if any of these show up in Hawaii, so that we can properly identify them, and then the genetic work can be helpful for management approaches in the future. The Alami babone story is a bit more complex, just because we still don't know exactly what causes it. And so we're in the business of building partnerships with some of the researchers over there where this disease occurs, and maybe trying to identify, we can't and won't do biological work here in Hawaii on this. It's just too much of a threat for somehow that you don't want it to escape. So whether it's a mainland location or somewhere else, where that work can be done. And we wanted to develop the diagnostics. And furthermore, I think identifying the insect vectors of some of these candidate viruses that are responsible for the disease, so that we know how it's spread and so forth. And I suspect we have the vectors of these viruses already here in Hawaii. So if they show up, I think they'll spread quickly. So that's all I've got. I want to thank a plethora of graduate students who have done work on this. Miriam Long, Ritten Gosai, he's now with Fiji, Biosecurity Authority, Jaron Laristo, Nelson Masang Jr. for collecting some of the samples in his home of Palau, Alejandro for doing a lot of the recent work, Dr. Wisen, former student here at UH, who's back in Papua New Guinea. He's the one who collected those samples for us, all of our members. And then just to point out, we did have permits for bringing this in, and no state permit was required because we brought all the samples in under preservative. So we only needed federal permits. So sorry, I think I went 5 minutes over what I wanted, but that's all I've got.
No problems. Thank you, Mike. So I have a question. So I used to hear about Daishin mosaic virus. Is that the same like a taro-vein chlorotic mosaic virus?
Different virus.
It's a different virus.
That one's been here for a long time. It's widespread. On a scale of 1 to 10, I typically give that one about a 3. it probably was very severe on some varieties, and so what happens, as we know, Hawaii had hundreds of different varieties developed by the Polynesians and subsequently, and probably when some of these new pathogens come in, they knock out the susceptible varieties, and all what's left are the ones that tend to be a bit more tolerant. And so that's probably why the taro varieties that are currently cultured tend to be a bit more tolerant to that virus. So I give that one about a 3 out of 10 in general in terms of severity. It's more of a nuisance than anything.
So all the virus you talked about, so Alamo bovine virus, it has maybe 3 different viruses?
We don't know. So that's the question.
Okay.
We know it needs more than one.
So it's all transmitted by insect vectors.
I think that's a safe bet. We don't know the insect vector, but I think that's a safe bet.
So both of these, Alamo bovine and taro beetle, we don't have yet in Hawaii, right?
Correct.
Just to make sure.
We do not have the beetle, and we do not have the disease, but we have potentially some of the components of the disease. So we have a few, some that might be involved, but again, since we don't know actually what combination actually results in the disease, we don't know if any of the viruses we have in Hawaii are actually responsible.
Okay, just in case, if the taro beetles will come to Hawaii, do we have any survey tool, like trapping pheromones or just like a coconut rhinoceros beetle traps?
There are no, not officially, although we do know that apparently they are attracted to a ricta lure, but I don't think we can do that. We are currently actually doing a survey for that beetle in Hawaii, where we anger taro growers by going around and like pulling up corms just to make sure. Obviously there's no evidence. We would know it if it was here. Let's put it this way. But we're still going through and doing a survey for that right now, as well as we're testing for some of those other viruses that were mentioned. And so far, everything's negative, aside for the ones that we already knew were here. We haven't found anything new.
So one more thing, just to make sure. So we can do diagnostic for Alamo bovine virus, right? That's what I.
No. Well, we can do some diagnostics, but so if you saw the Colocasia bobone associated virus, which is, I think, no question associated with the disease, we can diagnose at least 2 different strains of the virus, but there's so much genetic diversity that I'm not confident that we have a robust diagnostic. I'm not confident that if you gave me 10 infected samples or 10 symptomatic samples, maybe we would only detect the virus in 3 of them. Maybe we detect in 6. And that's not good enough when you need to detect them in everything. And so that's why we need more samples to understand the genetic diversity of the virus so that we can be more confident in our diagnostic ability.
Okay. Thank you. So let's check a chat box. So Surendra, I see. So where does that Bagrada bug came from?
It came from Africa.
Africa, okay.
Yes.
So I had a question in the chat box. So what I see here is they are, I mostly found them in a mustard.
That's right.
It's a mighty mustard. It's cover crop mustard or kai choy, pak choy, or in daikon. There is another associated question also. But in Maui, that's in Maui, they found everything is destroyed. So my question is, everything from radishes, any cruciferous plants or radishes, mostly maybe it's attracted more to radishes than other. So do they have any differential preference to the different cruciferous crop?
Not exactly. It all depends on how many are there, how healthy the crop is, and what stage. So I don't think anybody. I mean, there were studies that show a preference for several crops, various brassicas. But in what I heard from the growers is that there is nothing like that. They had kale, broccoli, and radishes, and everything equally infested. So lab studies could show certain things. So I was responding to that query that, one idea is to not grow them. Like, I mean, a commercial grower may or may not be able to avoid a crop like that, but, rotate with a non-host plant or use sweet alyssum as a trap crop, and then maybe spray something on it. Either a chemical pesticide if possible, or a biological pesticide that could probably help. And making sure that crop stays healthy is also important. They seem to be attracted more to weaker ones.
Mhm. So I think, yeah, you also answered here with sweet alyssum as a trap crop. So you mentioned about biological control for the Bagrada bug also, yeah? Is there anything that can be used here in Hawaii too?
There were some studies that they were doing, but I think they did not go to the extent of field releases because, the pest is no longer a big issue. So they didn't have enough to actually do something.
So another question is, why now Bagrada bug is not a big issue in California?
Yes, that's kind of a mystery. As I was, kind of alluding to it, we think it could be a very cold winter one season, 1 year.
Mhm.
That could have, knocked the populations down, and probably the fungi, because it is in the soil, like it tries to kind of hibernate in the soil or debris, not inside the soil, in the debris. Maybe the fungi, entomopathogenic fungi, provided some control, and then the native natural enemies also did it. And again, conventional growers have been able to effectively control with the chemical pesticides. So they reduce populations, and then temperature and all those things probably reduced it, suppressed it. But it was like almost like a mystery, like suddenly they disappeared. Like we were waiting to do more studies and figure something out, and then suddenly they disappeared. Only some organic growers see this issue in some areas, not all over.
So there is another question. Are there any pesticides that have been more effective than others for Bagrada, either conventional or organic?
Yeah, I think the conventional neonicotinoids and some other pesticides were very effective. I forgot the list because we don't recommend anymore, but there were some that worked very well. For biological and organic, I think pyrethrin, pyganic pyrethrin is one thing that commonly used, but I was mainly recommending, because I didn't have a chance, I was recommending azadirachtin and entomopathogenic fungi. That's what I was recommending at the time.
So entomopathogenic fungi will attack nymphs. That means nymphs.
That's
or
right.
adults?
Yeah.
Okay.
Well, when you use the combination, nymphs can escape the infection if they're just about to molt, and then you spray. They may not have the infection, so that's when you use this. Azadirachtin is a growth regulator and an insecticide and so on. So the combination can sometimes do better than fungus alone.
Okay, one last question. So we talked about the spotted lantern fly.
That's right.
So it looks like it's a pest of temperate fruit tree or other trees. So do you think of any potential host plants here in Hawaii?
Well, you have some grapes. I don't see. I checked before. I decided to talk about that. As far as I see, you don't have a lot of hosts, but you do have, like they can infest stone fruit, apples, grapes. So if you have them, and then I am not sure about the wild hosts. Do you have roses in Hawaii?
Yes.
Rose is a host. Yeah, rose is a host. So yeah, I think they do have some hosts that can support their populations. But when you have something like that, easy to identify, we could try our best to prevent the invasion.
Okay. Thank you. Any last minute question for Surendra as well as Mike? Okay, if there is no more questions, I would like to thank all the presenters. Thank you. I appreciate very much for your contribution in this mini conference. And I would like to thank all the participants and audience as well. Thank you for joining this mini conference. And I will be back in a few months with another mini conference. Thank you all.
Thank you,
And
Roshan.
I would like to say bye to everyone.